Thank you, Bear. Thank you to the organizing committee. Let's give them a round of applause. It's an amazing event, an amazing opportunity for all of us. And I'm just so happy to be here to share thoughts and ideas and to listen to Liam talk about purpose, to listen to Rodrigo talk about diversity and how we can share knowledge. So talk to one another, share these ideas. And to listen to Sarah talk about data. Sarah, I don't know where you are, but we need to talk. Remember, guys, data is at the very bottom of the pyramid of wisdom. Have you heard of it? So it's data, information, knowledge, wisdom. So she works with the very essence of wisdom. That is a pretty neat thing. I'm really happy to be here, but I have to leave tonight, so I'm actually sad because of that, because I want to hang out with you guys throughout the week. Actually, I have 3 little things to the organizing committee that I'd like to recommend. So the first one is to extend this event until the end of November instead of Wednesday. How'd you guys like that? And invite me. The second one is, instead of making it a biannual event, just make it an yearly event. And invite me, please. And the third one, and most important, always invite me to this event. I don't know if I've made myself clear, but... So we now are going to talk about a revolutionary technology, an innovative technology, that has the potential to change the genetic code. And it really resonates with every one of us, because we're all dependent upon DNA. So it's in our very essence. And this is a story about agriculture, obviously, a story about the gene, the DNA, but first and foremost, a story about our increased ability to improve the precision with which we can change the code of life. So agriculture started over 12,000 years ago, but our ancestors had no idea what the gene was, right? They couldn't go at the molecular level of life. But they could identify macroscopic characteristics that were interesting to agriculture and that helped them a lot. With, for instance, the adaptation to different settings. We were becoming settlers. We were establishing small communities. And we learned how to domesticate plants and animals, and that was a really good thing, because we were now able to produce bread, wine, beer. We were now able to improve the ways with which we did commerce, and this was really good for a very long time. But as I said, we were not understanding the molecular mechanisms behind life. That only came thousands and thousands of years afterwards with people like these guys. So Charles Darwin with the theory of evolution and natural selection, Gregor Mendel with a modern way to think about the science behind genetics. In the 20th century, we had a very important paper published in 1953 by Watson and Crick. I'm sure many of you have heard of it. So these two scientists proposed, alongside many others actually, proposed a model for the structure of the molecule of life. Why was this important? Because we were now able to create a whole new field within biology, within science. It was the recombinant DNA technology, our ability to use knowledge to rearrange DNA. And what did this lead to? So here we have an example. We were now able to get the human gene for insulin inserted into a bacteria, and now we could produce insulin and help with the lives of many, many millions of people who suffer from diabetes, just like we do it today. But we were also to get genes and insert them into plants, which happened in the 80s. And with that, we were able to generate plants that had interesting characteristics for agriculture. For instance, a herbicide-tolerant soybean, a soybean that thrives while pest plants perish. So I just gave you 12,000 years of the evolution of agriculture in 3 minutes. I know that's a lot. But now we're going to look at a common denominator that's throughout this history, and that is information. And guess what? Everyone here and everyone who is following us through stream is living in the information age. Isn't that so? So we now are able to obtain, to manipulate, to understand data in a way that has never before been possible. And the thing is that information usually comes in the form of a code. So the light that now hits my retina needs to be decoded, processed by my brain in order for me to understand the visual world around me. The voice that goes from my mouth into your ears needs to be decoded by your brain in order for you to understand what I'm saying. So information usually comes in the form of a code. And when we are able to get that code and apply it to generate products or services, we get technology. And is that different from biology? We know it's not. So biology also operates with a code, but instead of zeros and ones, it uses 4 letters, A, T, C, and G, in one of the main molecules, DNA. And those letters can be rearranged. They can form words, which are our genes. And when you compile enough words, you tell the story of a species. So how it is to be a soybean. What does make a soybean? You get a deck, a pile of genes that tell the story of the soybean. So now we're going to go through how these two fields are more connected than ever. So biotechnology, which we have been doing for thousands of years, is now at a modern stage, at the age of information. So we're going to take a look at how living information actually is processed within living organisms. So here is an example of different coding systems that your mobile devices can use. And just like your computer can analyze these data and transform it into characteristics, living organisms can do that with biomolecules. So these are examples of biomolecules that living organisms use as a type of decoding information. I already mentioned DNA, which is a major molecule in this process, RNA, a cousin molecule to DNA, and also proteins, the building blocks of life, are enzymes, structural proteins, hormones. And the thing is that the cell is able to interpret the code that is within the DNA molecule. It transcribes it into RNA. Then RNA can be read by the cell as well, and that is translated into proteins. And that gene information flow gives you a characteristic, the color of your eyes, how tall you are, or maybe the characteristics within a plant. So that is why if we change the information in the very essence, at the DNA level, we can change characteristics in the end. Well, but as I said, everything here is about coding. So this information is coded. And if I look at, let's say, a text from the Youth X Summit, it's also coded in the English language. But maybe I need to improve that information. What would I do? Because maybe I'd like to alter the code of a soybean. So is there an analogy that I can make? So for improving information in a text, I can use a text editor, right? So I could stumble upon a text like this with small information that needs to be changed, so a number that is not correct, maybe a part that is missing up, or a part that is added that shouldn't be there. What would I do? I would probably try to use a tool that is simple enough, precise, affordable, and also fast, so that I could, on a daily basis, change the text that I need to change. And by inserting what I need to find and what I need to have changed, I could tell this program to replace it by a more improved information, a correct information. So by the click of a button, I would ask for change a single number, maybe add a part that is missing, or maybe delete a part that shouldn't be there. And what scientists have done is they have created a tool that is able to do that with the genetic code, just like we do it with the English code in our computers. So we can now change a point molecule in the DNA. We can add a part that is missing or delete a segment that shouldn't be there. So in a very real way, we have reproduced what happens in a computer system that deals with coding and text editing, but now it is a molecular mechanism, a mechanism that changes molecules in living organisms. This is just one of the examples that you might have heard of. CRISPR-Cas9 is a very notorious system because of its characteristics, but it's not the only one, and we're going to talk about it in a little bit. And the thing is, it's also evolving. So just like agriculture, just like science or mankind, this is not the very first time we're talking about this. So gene editing has been on for quite some time now. The thing is, we have increased the feasibility through which we can do it. So initial tools, they cost a lot of money. They're really hard because you had to understand the physical-chemical aspects of proteins for each time that it needed to make a change in the DNA. And now with CRISPR, you can, from one single apparatus, make changes in virtually every single living organism in the planet, at least the ones that use DNA as their genetic material. So guess what? The latest tools, CRISPR-Cas, for instance, they are simple, precise, fast, and affordable. That is why we're using those, and that is why there's such a hype. Not only hype, but it's reality already. As Simon said, the future is here, and that is happening as we speak. So not only was this revolutionary in the context of a small or a single laboratory, but also Science magazine named it the 2015 Breakthrough of the Year. In order to get to that point, it needs to be something really substantial that would change science forever. And indeed, that's happening as we speak. And I'm going to go through it real quick. Okay, so for the ones who are not into molecular biology or who are not biologists, don't worry, this is really brief. So you have your double-stranded DNA, where the information, where the code of life is. With the help of a guide RNA, that cousin molecule to DNA, you can direct the location within the genome, within the pack of genes that you want the change to be made. And with a molecular scissor, for instance, Cas9, CRISPR-associated 9 protein, you can precisely change, or actually make a cut to the DNA, and that is the first step for you to start a changing process, either be a point mutation, deletion, insertion, and other types of change. The next slide, I don't need you to remember. So if there's a slide that you can forget, this is the one. I need you to understand the message behind it, the complexity behind it. So first off, you can see it's not only about CRISPR. There are different tools that you can use. The number of genetic modifications that you can do to the genome of almost every living organism in the world is pretty awesome. And this is only for traits in crops, as you can see up there. So this is just a small example from one paper that depicts different crops and the different types of change and characteristics that you have incorporated into them because of genetic modification at this level of precision. So, but what's in it for agriculture, really? Let's turn this into something more tangible. These are some of these characteristics. And if you look at them, they're either increasing yield, increasing productivity, reducing the need for natural resources, or actually improving final products for our consumption. So improving the oils in soybean, improving how branching occurs in tomatoes, and that helps with harvesting, protecting from diseases, herbicide tolerance, drought tolerance. That's all amazing. And again, as we look at the 17 objectives that we have for 2030, all of this is contributing a lot to many of them. So that is why this is an amazing tool, an amazing innovation that needs to be studied and understood by every leader of the future, because it's already happening. And I also encourage you to take a look at ISA's latest brief on how biotechnology is helping the world for over 2 decades to plant better, to grow better food. So we, with traditional modern biotechnology, we are using less pesticides, so less herbicides, fewer insecticides. We're producing more, increasing yield, so that helps farmers' gains. We're using less soil because we increase productivity, and therefore we can grow the same amount of food in a smaller scale. We're polluting less, so we're emitting fewer CO2 into the atmosphere. And more importantly, we're helping growers who are under terrible conditions today in developing countries to increase their gains and also alleviate poverty in other parts of the world that I need for people like you, people like us, who want to build a better world for agriculture and for everyone. So when I look at our ancestors and think about the things that they were doing and they were not even aware of, and honestly, we have accomplished a lot with artificial selection, with conventional breeding, I'm really optimistic because I'm sure that as we understand more about these tools and about the code of life, that we are going to be able to alleviate poverty, to eliminate hunger in a sustainable way for more and more people around the world. Thank you so much.
Dick. Just selfie. And Gustavo would like to do a selfie with you guys. Is that okay? Tutto ben? All right. Do we need delegates centralized here? Yes? All good? Are we all here? Yes.
Do you want to scooch up?
Stand up. I think you should stand up.
Okay, sure. Okay, awesome. Oh, absolutely.
I don't want to carry this up.
I hear you guys are fanatic about this stuff, so that's why I'm doing it. Okay, let's see. Okay.
1, 2, 3. Hey!
Awesome. Thank you.
Thank you. Okay. I think that presentation wins the prize for best presentation of data. Sorry, anyone else. That was incredible. I love that presentation as a data nerd myself. I suppose we've got lots of questions, yeah? Okay, so let's get cracking with that.
There's someone really excited right there.
Hi. So in Africa, we have a lot of examples of biotechnology that cannot be released because of anti-biotech laws. And a lot of these laws stem from specifically Europe's neo-colonial influence of the former colonies, not just in the soft power of us trying to emulate our previous colonizers' laws, but also in the hard power of European activists coming into our communities and actively campaigning against these technologies. What role do you see corporations playing in campaigning for evidence-based policy regarding biotech while still remaining ethical under late capitalism?
Starting with a big question.
Awesome question. Yeah. So communication, commitment to education, and the use of science behind it. So it is pretty clear that all of these technologies, and as I said, the most traditional modern biotechnology today is transgenesis. But now we have a new tool that doesn't necessarily lead to a transgenic crop at the end, and that helps with some regulatory hurdles. But really just to communicate the science behind that with a very efficient way, a way that connects with people, because there's so much data, as we're talking about data, there's so much data available showing us that this is positive. So we need to find a way to connect with people who still think this is harmful to the environment and to our health. And that's not the case. There's no rationale, scientific rationale behind this. So communication, communication, communication.
And so that's a big message. You as change makers as well, you'll be dealing with a lot of disinformation. There are things that are stated out there that are true, and there are things that aren't. And it's your job to take leadership with that. And that's really going to pass on to your whole generation. So that's an important point. Yes, another one.
Hi.
Hi.
My name is Mildred. I'm from Mexico, and I'm a biotechnology engineering student. And my question was actually very similar to his question, so I'm going to change it a little bit. But I've had the opportunity to be at the UN Biodiversity Conference a couple of times, and they, in the protocol of Cartagena, Nagoya, they regulate basically these applications. And for example, a couple of years ago, a moratorium on synthetic biology was proposed, and now they are trying to figure out what digital sequence information is, which is basically the nucleotide sequences, but they don't have a proper name for it. But we already have this infrastructure, like we have GenBank, and then companies do a lot of R&D, and so they're basically regulating the past. And I was gonna ask what he said, but now, can you talk to us, since you have a background on regulatory affairs, like how have you encountered these type of problems in your career? And I don't know, a little bit about your experience in this.
Right.
Okay, that's a great question.
Thank you for your question. So I think as scientists, sometimes we need to practice patience because science moves at a very fast-paced fashion, and regulations and the legal side of it just can't follow it, at least today with the mechanics of it. So if I have encountered issues regarding this specific aspect, but we need to understand that if we want to be the elements of change, we need to find a way to do that. So how is it that we scientists could influence regulators in order to, at a very positive way and at the most noble way of the word and sense of the word, of how to change the scenario and make sure that these regulatory processes are improved. It's not an easy task to be filled, but who said it would be easy? So, but with the use of science, I'm pretty sure that we can try to convince, but we need to use elements that will prove them that there's the rationale for us to continue doing biotechnology.
Okay, yes, we have a question over here.
Yes. Hello. Hi, Gustavo, here.
The guy
I'm
who's
Matteo.
really excited
Sorry,
about
Brazil.
asking questions. Okay.
First of all, don't leave the event without talking with me, okay? You are
I will.
prohibited. Don't
Okay,
go. Second of all, I'm fascinated with this recombinant technology of genes, but we cannot avoid public beliefs.
Okay.
Because in public, generally, there is this GMO controversy, which has nothing to do with science at all. We have compromised there's nothing, nothing wrong with GMOs, but people think when you annotate genes, you are going against the laws of nature, which is not true. But how we can, communicate with people in order to not have this Frankenstein perspective from people. How you do address these people in order to avoid such types of controversies.
Perfect.
Yes.
So it's another question about the disinformation, how we handle that.
Right.
Yeah.
So that's just interesting how this comes up.
Yeah.
So let me just ask, and I'm going to ask you a question now to the audience, and I need you to be very honest with your answer. How many of you here knew that transgenesis happens in nature naturally? Raise of hands. This is a biased audience, so a lot of people knew, but some of you didn't, and that's okay. And the fact is that the population all over the world, they don't know what DNA is, where it is, what it means to manipulate it, and they don't know that we have been changing the genetic code of several species since forever, since at least, some say 40,000 years before agriculture and us as settlers. So, and diversity and the natural selection process is dependent upon mutagenesis, is dependent upon change. So that has always happened. So try to break the barrier between natural and artificial. And why is it that natural is always better? If you get a mushroom and it's poisonous and you eat it, just because it's natural, it will kill you. And we have 2 decades of safe use of transgenic products, and we have not had a strong correlation with diseases or any condition. So the thing is, as I was speaking to some of you yesterday, we need to humanize science in a way. We need to connect with people at an emotional level to make sure that they understand what we're talking about, because showing graphs and tables and charts and numbers, that's not enough. So we need to create the environment and connection. So each one of you here can do that with your families. Why not? Why not start at home with friends, go out to bars and have discussions if they're into it? So there's several different ways, and we're definitely going to talk until today.
So there's part of, I think, one word to kind of encapsulate that as well is trust. What trust people have in science as well. Gene editing, yes, it's a scientific version of what farmers have been doing for millennia. I think we've got stuff we can learn from immunization, the fact that that's a scary thing to actually be given some sort of weakened version of a terrible virus, but then look at what's happened. We've eradicated smallpox. Mm-hmm. So these are the sorts of lessons we need to learn. We need to learn from what's come before, and we need to apply them in a new setting. One quick thing as well, with the squawk boxes, what we're going to do is we're going to actually throw them around. Who's into that? Yeah, so we don't select, you just throw it around,
Yeah.
and then it's going to be randomized, and then we'll really freak out whoever's talking. Okay, Courtney, do you have a question?
Good morning. My name is Courtney Davis. I'm the delegate from New Zealand this year, and I recently graduated with my master's in microbiology and virology, and I really relate to a lot of the things that you're talking about, and especially a lot of my fellow delegates' questions around science communication. I think it's really, really important to be able to tell a really correct and a really relevant narrative to the people around us in our communities and in our schools in particular. I'm really passionate about communicating to our young people, to our children in our primary schools, in our high schools, before getting to university, and I think this is a really, really important topic, especially around what Simon was saying about immunisation. There's a lot of scary science in the media. I was wondering if you had any advice or any ideas on specific examples of CRISPR-Cas and of gene editing and technologies like that, sort of examples relative to young people. So what kind of conversations, how could you have these discussions without talking about or introducing the ideas of recombinant DNA or genetic technologies like that? What are some basic concepts that you think we could discuss in our classrooms?
Right.
And just one point there, whatever you say to Courtney, she's going to take it back to New Zealand and start using it.
Oh, wow. Big responsibility.
She's going to profit from what you say.
And actually, I didn't answer. Rodrigo asked me to answer about nutrition and productivity. We can talk afterwards, if that's okay. So I have had the experience of going into schools and bringing insects that devour plants and actually compare conventional to transgenic plants and show how they can be how we can protect crops with biotechnology. So it was a very they could touch the plants, they could actually like grab the maggots and see how they behaved in their hands. So that brought the reality of agriculture into the classroom. And I remember that they were just astounded by what we could do with biotechnology because they saw it firsthand what was going on like in the field. And the thing is that especially for people who live in big cities, they have no idea what is agriculture. And kids are no different. So they think that the milk comes from the carton, like straight from the shelf. And that with many, many other products that we consume every day. So someone mentioned before about the gap that exists between growers and basically the rest of the population that is not growing, is not actually seeing the challenges that growers have. So how do you, again, interestingly enough, all of the 4 questions had to do with communication, but how do you connect with these people? This is about it, right? This is about showing the value of technology, of science, of tools like this to improve agriculture overall. So you need to connect. We can talk about several different ways afterwards, okay?
Okay, round of applause. Thank you so much, Gustavo.
Thank you. Mr. K? Obrigado.